Okay, let's dive in. We are tackling some really cutting-edge stuff in wildlife monitoring today, thinking about environmental nucleic acids.
Hmm.
And for years, the big player, the real star, has definitely been environmental DNA, right? eDNA.
Absolutely. Yeah, eDNA has, well, it's completely changed how we track life. Whether you're looking in seawater for whales or soil for fungi, even air for pathogens. eDNA lets you know if a species is present. It's finding those tiny genetic fragments left behind. Super powerful.
Hugely successful, yeah. But here's where we sort of hit a limit. If you're working in conservation, just knowing a species is there, well, that's often just the first step. Can eDNA tell you if that population is healthy? Is it like a thriving bullfrog nursery or just a couple of adults hanging out?
And that's the key limitation, isn't it? eDNA is like the blueprint. It's mostly static. It struggles to give you those crucial population details, demographics, age structure, that kind of status info.
Right.
And that gap is exactly what we're exploring today. We're focusing on a specific paper led by Megan B. Parsley. The title is Environmental RNA Can Distinguish Life Stages in Amphibian Populations.
Okay.
So our mission here is to understand how environmental RNA, eRNA, might give us that demographic insight that eDNA can't.
So let's get the basic biology straight first. DNA is the instruction manual. It's pretty fixed. Why does RNA, this eRNA, offer something more dynamic?
Okay, think of it like this. DNA is the big reference book in the library. It's basically the same whether you're larval or adult. RNA, though, is more like the immediate work order. It tells the cell, make this protein right now.
Ah, okay.
It reflects what genes are actually active differential gene expression.
Yeah.
And since an organism changes so much during its life, say tadpole to frog, the RNA signals change dramatically too. It's like a real-time activity log.
Got it. So we're shifting from just detecting the presence of a species to detecting its current biological state. So let's connect that to the real world. For amphibian conservation, why is it so critical to know if you're detecting a breeding population versus, say, just a few transient adults? How does that change things on the ground?
Well, it's fundamental for knowing if a population is actually viable, sustainable. If all you detect are adults, maybe year after year, that population isn't replacing itself. It's potentially aging out.
Right. No recruitment.
Exactly. And that information feeds directly into what conservationists call population viability analysis, or PVA.
TVA, that's a big one in conservation planning. So you're saying without something like eRNA, managers could be pouring resources into protecting a site, thinking it's a stable population, but in reality, it's just the last few adults fading away because the breeding habitat, the nursery, isn't working.
That's exactly the risk.
Yeah.
You absolutely need to know where the successful breeding is happening.
Yeah.
And amphibians are, well, they're kind of the perfect test case for this. They have these distinct life phases. They go through metamorphosis.
Right, the tadpole to frog transformation.
Yeah, a larva is so different, functionally, morphologically, from an adult. And those differences are driven by massive shifts in which genes are turned on or off.
Which means the RNA signature should be drastically different. Okay, so the researchers focused on two species here. The American bullfrog, Lithobates catspianus, and the tiger salamander, Ambystoma mavosium.
Correct. And their goal was to find a molecular marker, like a specific signal that only the larvae produce. So they developed these gene-specific eRNA assays targeting genes expressed only in that larval stage.
Okay, what genes?
They honed in on keratin-associated genes, specifically RON-R larval keratin, RLK for the bullfrogs, and keratin 6A, K6A for the salamanders.
Keratin. Now, I usually associate keratin with tough stuff, like adult skin or hair and nails in mammals. Why target keratin for a larva?
But it's about the change. These specific keratin genes are heavily involved in remodeling the skin during metamorphosis. You're going from that soft, permeable larval skin, tadpole skin, to the tougher, more protective adult skin. This involves switching specific larval keratin genes on, and then crucially, switching them off later. While the larva is developing, it's actively expressing and probably shedding cells containing this larval keratin RNA into the water,
making it a detectable eRNA signal, a unique temporary signal.
Exactly. A powerful, stage-specific signal.
Okay, so they identified their targets, RLK for bullfrogs, K6A for salamanders. But before taking this out into messy real-world ponds, they needed to be absolutely sure these signals were truly larval-specific in the lab. How'd they do that? You mentioned a three-step process.
Yeah, they were really rigorous. Basically, 3 levels of testing. Step 1, biological specificity. They took direct skin samples from individual animals at different life stages. They had to prove, right there in the tissue, that the gene expression was genuinely exclusive to larvae.
Okay, makes sense. Tissue first, then what?
Step 2. Environmental relevance. They checked if that same pattern held up in the water samples taken from the tanks where the animals were housed.
Right, because water gets stuff from skin, waste, everything.
Exactly. It's a more complex mix. And step 3 was about consistency. They checked the pattern over time within a single developmental stage just to make sure it was stable. It was quite thorough, over 220 samples across both species in the lab phase.
And how did the assays perform in these controlled lab tests? Were they sensitive enough?
Oh, they worked really well in the lab. Detection rates for the larval RNA were high. For the bullfrog RLK assay, they got 90% sensitivity in the larval samples.
Pretty good.
And the salamander K6A assay was similar, 88.4% detection in their larval samples. So it definitely confirms that larvae are shedding enough of this specific RNA to be readily detectable in their immediate environment.
Okay, sounds great. But you mentioned earlier there was a slight wrinkle, something unexpected with the salamander K6A assay in the lab.
Yeah, this is an interesting and important detail. While the K6A RNA was totally undetectable in the adult salamander skin tissue itself, they found some very low-level, sort of non-specific amplification of K6A in the water samples from the adult salamander tanks.
Huh. So a tiny bit of the larval signal showing up in the adult's water, even though it wasn't in their skin. That sounds problematic, like it might not be perfectly specific after all.
It did definitely raised a flag. It suggested that maybe, just maybe, this gene was expressed at very low levels in some other adult tissue, or maybe released through waste, perhaps.
Right.
They acknowledged this, but the thinking was, okay, in a controlled small tank, maybe you pick up this faint background noise. But out in a large, complex, natural pond or wetland with huge dilution, maybe this super low-level signal wouldn't actually interfere with detecting the real larval signal.
Okay, so it's a question of scale and signal to noise. Does a tiny lab artifact matter in the wild? We'll get to that field test soon. But first, they tackled a huge practical issue. RNA is notoriously fragile, much more so than DNA.
Oh, absolutely.
If you have to flash freeze every water sample immediately in the field, that makes the whole method way less practical, especially in remote areas.
Exactly. DNA is tough. RNA breaks down easily. This is a major hurdle for eRNA work. But they showed they could successfully preserve the eRNA in field samples without needing immediate freezing. They used a stabilizing solution, DNRNA Shield, and found they could store samples at room temperature for up to a month without significant degradation.
That's huge. That makes field collection actually feasible for regular conservation teams.
It really is a game changer for deploying eRNA methods widely.
All right, so they have validated assays, a way to preserve samples. Time for the real tests. Natural ponds and wetlands. What happened with the bullfrogs out there?
For the bullfrogs, using the RLK assay, they had pretty good success. They detected larval RNA in about 74.1% of the ponds where they knew larvae were present.
Okay, solid field detection. But you said the salamanders offered a particularly neat test of specificity.
Yes, the long-toed salamanders, A. macrodactylum, were key here. Their life cycle in the study area created a kind of natural experiment. See, the adults migrate to these ponds to breed in the early spring around March, and they leave. The larvae develop over the summer.
Ah, so you have distinct times when only adults are present, and later only larvae.
Exactly. They could sample the water in March, when just the breeding adults were there, shedding DNA and whatever else, and then they could go back to the very same ponds in June, when the adults were long gone, and only larvae remained.
Perfect setup. So, the moment of truth. What did the larval-specific K6A RNA assay show in March versus June?
This was the really crucial finding. When only larvae were present in June, the K6A RNA detection worked well, about 70.8% positive detection overall.
Okay.
But, and this is the key point during the March sampling, when only adults were present, even though they were actively breeding, and eDNA detection was high.
Yeah.
The larval-specific K6A RNA was not detected. In any PCR replicate, 0.
Wow. Zero detection of the larval marker when only adults were around, despite adults clearly being there and shedding DNA.
Exactly. That's incredibly strong evidence for the assay's life stage specificity out there in the wild.
And what does that say about that low-level noise they saw in the lab tanks?
It strongly suggests that their hypothesis was right. Whatever tiny bit of non-specific signal might exist in a controlled lab setting, it just doesn't seem to interfere or cause false positives in a real, complex, large-volume natural system. It really confirms that this RNA approach can reliably distinguish life stages for practical management.
That's fantastic. Translating the molecular detail into a clear field result. What about comparing RNA detection rates to standard eDNA overall in the field? Was RNA as sensitive?
Generally, the detection rates for RNA were a bit lower than for the established eDNA assays they ran alongside it. And interestingly, for both RNA and eDNA, detection rates were lower at sites where the researchers just qualitatively, by observation, thought the population densities seemed lower.
Okay, so RNA is specific, but like eDNA, it's still harder to detect when population numbers are very low. Makes sense.
Yeah, the basic challenge of finding a faint signal in a big environment still applies. But let's really emphasize the novelty here. This study isn't just another detection method. It's considered the first successful use of ERN analysis to get population demographic information, specifically life stage data from natural populations of macroorganisms, like vertebrates.
That's a pretty big first step.
It really is breaking new ground.
So thinking about the immediate impact, how can conservation managers use this, like right now?
Well, these kinds of assays could be deployed relatively quickly to pinpoint those critical active breeding sites in nursery habitats.
Yeah.
As we discussed, knowing where successful reproduction is happening is vital for effective management, for prioritizing conservation actions, and for building better population models, like those PVAs.
Focus the resources where they matter most on the nurseries.
Okay, but it can't be all smooth sailing. What are the main hurdles or challenges holding eRNA back from being as common as eDNA, perhaps? There are definitely challenges. First, there's still a pretty big knowledge gap. We know a lot about eDNA sampling, filtering, how it behaves in the environment. For eRNA, we need much more basic research, like optimal sampling methods, the best ways to filter and process samples for RNA, understanding its state, fate, and transport. How long does this eRNA signal actually last in different conditions? Cold water versus warm water, soil versus sediment.
Right, the fundamental environmental behavior. What else?
Second is moving beyond just presence-absence. The next big step is refining the quantitative side, getting really accurate limits of detection, limits of quantification.
Yeah.
The goal isn't just, yes, no, larvae are here, but ideally estimating the proportion of larvae versus adults based on the eRNA signals.
That would be powerful, getting actual age structure data from a water sample.
Exactly. That's sort of the holy grail for non-invasive demographics.
And what about actually developing these assays in the first place? Is that easy?
That's the third challenge. Developing these highly specific assays relies on having good genetic information, specifically transcriptomic data, knowing which genes are expressed when. For many species we care about, especially non-model organisms, we just don't have robust transcriptomic resources. That's a bottleneck.
Although the paper suggested a potential workaround.
Yeah, they mentioned that we might be able to leverage the extensive data available for closely related species like well-studied model organisms to help predict and design these stage-specific markers, even if we lack data for the exact target species. It's a potential shortcut.
Okay, so zooming out to the big picture then, environmental nucleic acids are clearly continuing to revolutionize monitoring. eDNA gave us presence-absence, and now eRNA seems poised to add this whole new layer of resolution, getting at population status and demographics.
That's the core message.
Yeah.
eRNA opens up this incredibly rich source of biological information. We're moving beyond just knowing if a species is present to starting to understand the state of that population. Is it actively breeding? Is it viable? That's a major leap.
So let's boil it down for you listening. This work suggests we're getting closer to a future where basically a conservationist could take a scoop of pond water and not just find out if an endangered frog lives there, but get a strong clue if that pond is actually serving as a vital nursery producing the next generation, or if it's just a stopover for adults. That's a huge difference for where you put your conservation dollars.
It really is. And it makes you wonder, doesn't it? Given this success with amphibians and their complex life cycles, what other organisms could really benefit? Think about marine invertebrates with larval stages, or migratory fish with distinct freshwater and saltwater phases.
Oh yeah, tracking spawning runs or settlement of larvae.
Exactly. Where could tracking ERA provide the most critical insights into these complex life histories? That's a really exciting avenue for future work.
The potential to monitor the biological activity of populations non-invasively across ecosystems.
Yeah, that's definitely something to think about. A fascinating glimpse into the future of biomonitoring.